Hello children, welcome to Vivid Botany. We have a question here about the vector as well as the sequence that controls the copy number of linked DNA. To know about the question, we should discuss something quite interesting. Let us see that this is a bacteria, right? So in this bacteria, its own DNA is present, which is double stranded circular naked DNA is present. So in this bacteria, we have introduced a DNA. We have introduced a dsDNA. This is a foreign DNA introduced into the bacteria. Now the bacteria is let to undergo binary fission to form two bacteria. Two daughter bacteria are formed here. Now in two daughter bacteria, we will see that whether the inserted foreign DNA is inherited or not. So in this, the foreign DNA is not inherited. I write here, foreign DNA is not inherited. Yes. Why the foreign DNA is not inherited? That is the question here. Now, in experiment 2, let us take it as experiment 1. Let's take it as experiment 2. In this experiment, what do we do that? Here, also we take the bacteria, and in this bacteria, we insert the foreign DNA inside its genophore. We insert the foreign DNA in its genophore. Let us suppose this is the genophore we have inserted here. So this is the foreign DNA in which we have inserted it. Now we let it undergo the binary fission that gives rise to two daughter bacteria. So in daughter bacteria, we see that whether that foreign DNA is inherited or not. Now surprisingly, we understood that foreign DNA is inherited here. In this daughter bacteria also inherited. In this also it is inherited. So why in experiment 1 foreign DNA is not inherited, and why in experiment 2 foreign DNA is inherited? That is the thing we have to understand. Now let us see that experiment 3. In experiment 3 also again we take a bacteria with its own DNA, double stranded circular naked DNA, and this time we insert the DNA that is having ORI. This is the foreign DNA that is having ORI. So let me write this is foreign DNA with ORI. Now, when this foreign DNA with ORI, remember ORI stands for Origin of Replication. It stands for Origin of Replication, and now it is inserted inside the bacteria along with ORI. Okay. Now we allow the bacteria to undergo binary fission to form two daughter bacteria. And two daughter bacteria, we check that whether this foreign DNA along with ORI is inherited or not. Yes, here also the foreign DNA along with ORI is inherited. So, foreign DNA along with ORI, that is origin of replication, is inherited. That is the case here. Now, what happened in first case? that why foreign DNA is not inherited here. And in second case, why foreign DNA is inherited. In third case, why foreign DNA is inherited. Remember one thing that when ORI is present, the site responsible for origin of replication, then only the replication will start, and that is inherited into the daughter bacteria. We know that without replication, two copies are not formed from one copy. They are formed only with replication, and replication is done only with ORI only. So when that ORI is present, it is getting replicated and it is getting inherited to daughter bacteria. In the first case, the foreign DNA, remember this is without ORI. So it is not inherited to the daughter bacteria. In second case, the foreign DNA, foreign DNA is also without ORI. This is also foreign DNA without ORI. Okay, so as the foreign DNA is without ORI, but even then it is inherited. Why? Yes, why it is inherited without ORI? Remember, the main important point you have to understand here is that ORI is not present with foreign DNA, but foreign DNA is integrated with bacterial DNA which is having ORI. Hope you have understood. In second case, foreign DNA is not with ORI, but in second case, foreign DNA is integrated, attached to bacterial DNA that is with ORI. So that in second case, it is being inherited, and in first case, foreign DNA is not inherited because that bacterial DNA is having ORI, but foreign DNA is not integrated with bacterial DNA. The foreign DNA remains isolated in bacteria. It is not coupled with bacterial DNA and not having the ORI. So, you should remember one thing that either the foreign DNA should have ORI sequence or the foreign DNA should be integrated with bacterial DNA that is having the ORI. So ORI is compulsory for replication of DNA. Without ORI, it cannot be replicated. Of course, it can be replicated, but when it is attached to the bacterial DNA that is having the ORI. And in third case, you see, bacterial DNA, of course, it is having ORI, and foreign DNA also having ORI. So foreign DNA, though it is not integrated with bacterial DNA, it is being replicated. Hope you have understood. Though the foreign DNA not having ORI, so foreign DNA is having ORI and not attached with bacterial DNA, but even then it is replicated. The reason is that this foreign DNA is having here ORI, that is origin of replication. Now let us see the question here, that the sequence that controls copy number of linked DNA with the vector is termed as. Yes, the answer is here ORI. Yes, so ORI site not only what do you call ORI site is not only responsible for origin of replication, but it also controls the copy number of foreign DNA in host cell. So, it also controls the copy number of foreign DNA in host cell. That is, you need to understand. That is, for example, There are two ORIs are there. This is one ORI. Let's name it as A. This is one another ORI. Let's name it as B. So ORI A will make up, let us suppose, 15 copies, whereas ORI B will make up 20 copies. ORI B will make up 20 copies. ORI A will make 15 copies. So which ORI do we select here? We select ORI B. Why do we select the ORI B? Because this ORI is having 20 copies. It can support 20 copies. Remember, the ultimate aim of the plasmid, the recombinant DNA technology, is that to get as many copies as we can through the host plant, host cell. So we should select that ORI which can support maximum number of copies. So in case B, which can support 20 copies, in case A, which can support 15 copies. So that is the difference we find here. So ORI not only responsible for initiation of replication of the vector, but also it can support the number of copies present in that host cell. So it is a very good question that he is asking the additional functionality of the ORI, because everyone knows that ORI meant for origin of replication and ORI is responsible for initiation of replication of DNA. But here he is asking the additional function of the ORI, that is controlling number of copies in host plant or host animal. So the answer is 2. Hope you have understood the explanation that is given for the question. It is taken from biotechnology principles and process. So I hope you are preparing very well. You have any query, any doubt, any clarification regarding the concept, mail me and just put up in the comment box. We are ready to explain you further in depth about the concept. So we are discussing from Vivid Botany. Please subscribe the channel Vivid Botany. Thank you very much.